Folks, I got everyone here. Thank you for coming. In case you didn't figure it out, we're here for the thesis defense of Eiren Jacobson. If you thought it was somebody else, it's easy. I'm Peter Franks. I'm Eiren's co-advisor of Jay Barlow, and I'm just here to tell you the procedure. So Eiren is going to give a presentation after Jay. It'll last on the order of 50 minutes or so. After that, we have public questioning, so all of you can take the nastiest, most technical questions and ask Eiren. And then we will shoo you out, and you can go over to Surfside and hang out. Surfside is that way. And the committee will ask Eiren dark penetrating questions, and then we will hopefully be signing papers with yeses and signatures. And then we will celebrate over in Surfside, and the food arrives, I guess, at 11:45. So, yes, we'll be very much. All right, I'll turn it over to Jay.
Okay. It's my great honor to co-chair the dissertation with Peter, and actually with Karen Forney too, because Karen Forney is the unofficial chair of Eiren's committee. She's been Eiren's mentor and field assistant and general do everything person for her, and made Frank's and my job very easy. So the pictures you see here are Eiren deploying her own equipment, but what she won't have time to talk about was that she was actually, during her relatively short tenure here at Scripps, able to participate in lots of other field projects as well. In fact, she started to work for me before she actually entered graduate school, doing acoustic work on a cruise to Hawaii. She's been on West Coast NOAA cruises. She's been on several of the behavioral response studies that we've done in Southern California. And so she's always jumped at the opportunity, and I don't know if you've ever tallied how many months you have spent at sea, but it certainly exceeds the minimum requirement for Scripps graduation. As I mentioned, her stay here has been incredibly rapid from my perspective. 5 years. Your parents are probably saying, "What took you so long?" But 5 years is really short. And and in that time, she has 3 of her dissertation chapters already published, and she had a publication before that on some of the work she did for me prior to that. So she's done a really good job at getting those publications out. She's also been able to travel quite a bit as a graduate student. The top picture is her in Akaroa in New Zealand. So she's been to 2 marine mammal conferences of the Society for Marine Mammalogy, 2 conferences in Europe for the European Cetacean Society, and a conference in Seattle where she actually was awarded the Best Student Paper Presentation prize at that meeting. So without further ado, Eiren.
All right, good morning, everyone. Can you hear me? Yes, okay. Thank you all so much for being here today. I'm really excited to see so many friendly faces in the audience. I'm really looking forward to telling you about the work I've done as part of my dissertation, but first I'd like to thank my committee, including my co-chairs, Jay Barlow and Peter Franks, who you both met, and also Elsa Cleland, Karen Forney, Jules Jaffe, and Bryce Simmons for all of the guidance and support they've provided to me over the years. I'm so grateful for the graduate education I received here at Scripps Oceanography, and I'd also like to acknowledge the important role the NOAA Southwest Fisheries Science Center and Moss Landing Marine Labs played in supporting my dissertation research. So today I'll be telling you about a few different projects I've worked on over the past 5 years that all fall under the umbrella of developing a passive acoustic monitoring network for harbor porpoise here in California. So first, let's talk a little bit about why we monitor animal populations. There are a lot of different reasons. I'm just going to highlight a few. For example, monitoring animal populations can allow us to identify species of concern So for bald eagles, the pesticide DDT has unintended consequences. Monitoring the populations, we wouldn't have realized that it was having a negative effect on bald eagle populations. We can also use monitoring to evaluate conservation actions. So if we install fish ladders to help salmon get upstream, we can use monitoring to help determine, is that effective? Is that making a difference? For animals that we hunt or fish, monitoring allows us to determine allowable take. So basically, how many animals can we safely take out of the population through hunting or fishing without causing the population to collapse? For marine mammals, there's another reason, and that is that it is mandated by Congress that all marine mammals within U.S. waters be monitored by federal agencies. That as part of the Marine Mammal Protection Act. And so this includes everything from sea otters to whales and dolphins to manatees, all marine mammals found in the United States. One of the ways that we monitor marine mammals is using what are called line transect visual surveys. These surveys use an airplane or a large ship as a platform, and visual observers, like this one here, scan the sea surface and count the number of animals that they see. We then use statistical methods to basically extrapolate that count to the total number of animals present. Using line transect visual surveys, we can estimate both density and abundance. So just to explain what I mean by that, when I say density, I'm referring to the number of animals per area. So typically animals per square kilometer. Abundance is the total number of animals. For example, 100 whales. So there are advantages and disadvantages to using line transect visual surveys. Some of the advantages are that if you're using a ship or an airplane, you can cover a large swath of the ocean, so you can get a large geographic range. Also, because you're using visual observers, you can confirm species identification, so distinguish closely related species of dolphins, for example. Also, these methods have been developed over the past several decades, so there are established statistical techniques for estimating density and abundance from visual survey data. Some of the disadvantages are that marine mammals, being marine mammals, spend a lot of time underwater. So particularly for some deep diving whale species, it can be difficult to get accurate estimates of density and abundance because they're not available to be observed most of the time. Visual surveys are also limited by weather. So anything from fog to wind to rain can make it difficult or impossible to conduct visual surveys. The platforms, like big ships and airplanes, tend to be expensive to operate, and therefore, because of limited budgets, the surveys are conducted fairly infrequently. And this is particular to aerial surveys. Flying in a small plane over the ocean poses a risk to both the pilot and the observers. So people have been interested in other ways to monitor marine mammals, and one of the methods that's gaining a lot of popularity is passive acoustic monitoring. So passive acoustic monitoring relies on listening passively, so with an underwater microphone, to the sounds that marine mammals make. So they make sounds to navigate, to communicate, to look for food. We basically eavesdrop on those sounds and use them as a proxy for the animals themselves. So this is usually the acronym PAM, passive acoustic monitoring, has been used to monitor cetaceans, including fin whales, sperm whales, and beaked whales, and cetaceans are whales, dolphins, and porpoises. So now that I've told you a little bit about passive acoustic monitoring in general, first, there are advantages and disadvantages to this method also. So some of the advantages are that sound can travel long distances in the ocean, so you can hear animals that are quite far away, as opposed to visual surveys, which are limited by the distance you can see. It's possible to use passive acoustic surveys to do long-term monitoring. So once you put an underwater microphone out, you can leave it there for months or even years and get long-term time series of animal presence. Passive acoustic surveys tend to not be weather dependent. So again, once you have an instrument deployed, it doesn't matter if it's foggy or rainy, you'll still be collecting data. And another advantage is that it's a non-invasive method, so it's not impacting the animals. Some of the disadvantages are that sound can travel long distances in the ocean. So it's hard to know how far away the animal is sometimes. And that can make it tricky. Also, the ocean is noisy, both with other animals, things like snapping shrimp, fish make noise, but also humans make a lot of noise in the ocean, so that can interfere with passive acoustic monitoring. Going along with that, a lot of species make very similar vocalizations. So especially closely related dolphins have very similar whistles, and it can be hard to tell like which species it is that you're hearing. And finally, call rates are often unknown. So we don't know a lot about how often animals vocalize or what affects how often they vocalize. So now that I've told you a little bit about passive acoustic monitoring, I'm going to tell you about the species that I studied in my dissertation. So I focused on the harbor porpoise, which has a nearshore distribution in cold water of the northern hemisphere. So that's shown this light blue shading on this map. So you see they're found in northern Europe, along the east coast, and along the California coast up into Alaska. They're a fairly small marine mammal, so they're about human size. They're about 5 to 6 feet long, 150 pounds. Wild, it will look like this. So it has a very distinctive triangular dorsal fin, and it sort of just flips above the surface of the water very quickly. They're not a social cetacean, so they don't form stable family groups. They don't really hang out together. They tend to be in groups of 1 to 3. And luckily for us, they use echolocation to navigate and to forage for food. And they only use echolocation. So unlike some dolphins that also whistle and sort of communicate that way, they only use echolocation clicks. Their echolocation clicks are extremely directional. So you can think of it like a beam of sound, like a flashlight in the ocean, sweeping across the seafloor. And so that's what's shown in this diagram here. So this is just the intensity of sound as reported on hydrophones around an animal that's echolocating, if the animal were right here and facing up. So the more red is louder, is more intense sound than the blue areas. And you can see that it's just a cone right in front of the animal. Directly behind the animal, there's almost no awful sound. And then that's the same idea shown here. So this is a porpoise, and again, just has this cone of echolocation propagating forward. Their echolocation clicks are described as being narrow band and high frequency, and they tend to travel hundreds of meters. So they're what's called their acoustically active space. The space that they're echolocating is on the order of hundreds. So just to put this in perspective, when I say high frequency, so this is just an axis of frequency. So this is how basically pitch. So low frequency is deeper sound, and high frequency, higher sounds. Okay, so here's humans. This is us. We typically vocalize between 1 and 4 kilohertz. That's this dark green area. And we can hear, at least when we're young and female, up to about 20 kilohertz.
Read my mind. You got a man there, though.
It's ambiguous. Okay, so that's us. Harbor porpoise, on the other hand, they echolocate at about 135 kilohertz. And they can hear sounds from about 15 to 140 kilohertz. So the echolocation clicks that they make are so far outside our range of hearing. There's no way to be able to hear them. And interestingly, they are not the only animal to use this kind of echolocation signal. So there are other porpoises like Dall's porpoise, but also species like pygmy and sperm whale, and some small dolphins that make almost identical echolocation clicks. And this evolved independently in multiple different lineages of mostly small-bodied cetaceans. So one hypothesis for why they've all converged on this very similar echolocation signal is to avoid being heard by killer whales. So killer whales like to eat small marine mammals. And so one idea is that they've evolved an echolocation signal high enough that it's outside the hearing range of killer whales. So passive acoustic monitoring has been used for harbor porpoise mostly in Northern Europe. This is a map of the Baltic Sea, and all of the red dots are passive acoustic sensors that were deployed there. It's also been used to monitor the critically endangered vaquita, which is a close relative of the harbor porpoise, in the Gulf of California. So this is the northern Gulf of California, and all of the dots are passive acoustic sensors. There are some challenges remaining, some of which I addressed in my dissertation, including, as I mentioned, there are multiple species that make very similar echolocation clicks. So one challenge is being able to discriminate those clicks between species. Estimating absolute abundance of animals as opposed to relative abundance or trends is still a challenge. And finally, there's not been a lot of work on optimizing the survey design of passive acoustic studies. So in my dissertation, the main objective was to use a specific population of harbor porpoise as sort of a case study for developing passive acoustic monitoring methods. And I did this, I had 4 data chapters and 1 introductory chapter that was more of a review. So my data chapters in chapter 2, I used passive acoustic sensors to document interactions between harbor porpoise and bottlenose dolphins in Monterey Bay, and found that harbor porpoise were avoiding bottlenose dolphins in that region. And that study was published in 2015 in Marine Mammal Science. In chapter 3, I recorded harbor porpoise echolocation on 2 different types of passive acoustic sensors, clicks and the peak frequency of clicks recorded on the 2 different sensors, with the idea of determining what kind of sensors would be able to discriminate the narrowband high frequency clicks of closely related species. That study was published as a NOAA technical memorandum this summer. In chapter 4, I used paired visual, so aerial surveys and passive acoustic surveys to estimate the detectability of harbor porpoise by passive acoustic sensors. That was published in the Journal of the Acoustical Society of America earlier this year. And finally, in chapter 5, I used a simulation framework to investigate different possible passive acoustic network survey designs. So because our time here today is limited, I'm only going to be talking about the last 2 chapters of my dissertation in detail. So I've given you a little bit of an introduction to harbor porpoise and to passive acoustic monitoring. Next, I'll describe both the aerial survey and the passive acoustic methods that I use that are relevant to both of the chapters I'll be talking about. Then I'll describe how we used that paired data set to estimate detectability of harbor porpoise. And then describe the simulation that I used to look at different designs of passive acoustic surveys. And I'll wrap up with some conclusions. So methods. So like I said, I used the Monterey Bay population as a case study. So I was looking specifically at California. Here in California, we have 4 distinct populations of harbor porpoise. There's a Southern California, or Northern California, Southern Oregon population, a San Francisco Russian River population, a Monterey Bay population, and a Morro Bay population. Harbor porpoise tend to be found in fairly shallow water, less than about 200 meters deep. And because the continental shelf off of California is so steep, that results in a very near shore distribution. So that's shown in this sort of teal color. That's the distribution of porpoise. And since the 1980s, these populations have been monitored using aerial surveys conducted by the NOAA Southwest Fisheries Science Center. The Monterey Bay population specifically is about 3,700 individuals. That estimate comes from aerial surveys, and the confidence interval on that is 1,500 to 9,500. Again, they're found in fairly shallow water, so this light purple is showing the distribution out to about 200 meters, and then their highest densities tend to be in water about 10 to 40 meters deep. And again, we know that from aerial survey observations. Some recent work by Karen Forney, who's on my committee, combining aerial surveys conducted over a 25-year period, has found that the population is probably increasing. So this is just showing a time series from the late 80s to 2013 of harbor porpoise abundance in Monterey Bay. And you see that it's trending up over time, albeit with large confidence intervals. Okay, so the aerial surveys the data from aerial surveys that I'm using in my dissertation were collected using a Partenavia P-68 twin-engine aircraft. So these are 2 of the planes that we use to fly aerial surveys. You see that they have these high wings, so you can see out the windows, you can see out and down into the water. This is what it looks like inside the plane. These are pretty old planes. So there's a pilot and a data recorder sitting in the front. And then in the back, we have 3 observers who are all here today. Thanks, guys. So 2 observers look out the side of the aircraft and down, and then 1 grumpy observer in the back. There's in the back. There's actually a hole in the bottom of the airplane with a plexiglass window, and so you look down through that window to count the animals that are directly below the airplane. So specifically for my dissertation research, we did aerial surveys on 4 days in 2013, which resulted in 2,600 kilometers of aerial survey effort in the Monterey Bay region, and that's shown in the black lines here. So we surveyed the northern part quite a bit and then did 1 survey of the whole stock range. Luckily for me, I had access to this amazing data set collected by Southwest Fisheries over decades, and I used data from 2000 to 2013, when about 26,000 kilometers of aerial survey effort were flown. So that's 10 times more data than I collected specifically from my dissertation. Similarly, we deployed passive acoustic sensors in the northern part of Monterey Bay. The sensor that I used is called a CPOD. It's manufactured by a company in the UK. So it consists of an underwater microphone called a hydrophone with an onboard echolocation click detection algorithm. So it stores information about each click that it hears, but doesn't record full bandwidth sound files. And that's to save space, basically. After recovering the instruments, clicks are classified into what are called echolocation click trains, which are just series of clicks. Using an algorithm that is proprietary to this particular sensor. So I deployed 11 of these for 3 years in the fall of 2013, 14, and 15, and these are the locations of the sensors in the northern part of the bay. This is what they look like. I think Jay might have shown this picture. So these are tubs, and each tub is 1 sensor with all of its mooring equipment ready to go. This is what the CPOD itself looks like. And some of our CPODs were deployed using scientific divers from Moss Landing Marine Labs who helped us with the installation and retrieval. And some were used acoustic releases. So here's a diagram of what we call the deep design. So these were put in water about 30 to 75 meters deep. We had a cement anchor, a sacrificial anchor on the bottom, an acoustic release, and then a hard float that suspended the CPOD into the water column. So basically, when we wanted to get them back, we would trigger this release and it would float to the surface, leaving the anchor on the seafloor. For our shallower water designs, we used a design that included a surface marker. So here we have 2 floats, a weight, and then a chain to a sand anchor that allowed the mooring to swivel. And these were installed, the shallow water moorings were installed by divers. Once we got the sensors back, the data is actually stored on 4 gig SD cards, just like we'd use in your camera. And it's all, like I said, it's not storing waveform data. So it's just, it gives you a record of each echolocation click that it heard. So that's what the data look like when they come out. And from that, I then process the data into a usable metric. And the metric I used is the number of porpoise positive seconds. So the number of seconds in which a porpoise was detected. The advantages to this, as opposed to using the absolute number of echolocation clicks, are that it reduces the impact of orientation on detectability. So because harbor porpoises are sweeping their beam by averaging over a 1 second period, it reduces the influence of that sweep. And also using seconds as opposed to minutes or hours, the metric is less likely to be saturated. This metric does assume that only 1 animal can be detected within each 1 second period. Okay, so now that we've gone through some of the methods, let's talk about the specific chapters. The main objective of chapter 4 was basically, okay, so we hear some echolocation clicks, but what we really want to know is how many animals are there. So how do we get from the number of echolocation clicks that we hear to the number of animals that are present? So schematically, this is what we're doing. So we record some clicks. We want to know how many harbor porpoise there are. So we need something to multiply the number of clicks by to get to the number of harbor porpoise. So here are animals clicking. One thing we need to know is how often do they click? So what is the probability of echolocating? So that we can adjust for that. The other thing we need to know is the probability that those clicks are detected by our passive acoustic sensor. So you can imagine that animals that are further away from the sensor are less likely to be detected than animals that are close to the sensor. And similarly, animals that are facing away from the sensor are less likely to be detected than animals that are facing towards it. So we can describe the relationship between distance and the probability of detection using what's called the detection function. So this is just showing distance on the x-axis and the probability of detection on the y-axis. And as you would expect, you're much more likely to detect animals that are close than animals that are further away. From this detection function, we can calculate what's called the effective detection area or the effective detection radius. So that's the area around the sensor where you're able to detect animals. Okay, so these are the two things we need to know in order to get from the number of clicks to the number of harbor porpoise. Because of the nature of the data set that I have, I'm not going to be able to estimate these two things individually. So I'm just going to refer to them jointly as detectability. So detectability, I'm calling detectability the product of the probability of clicking and the effective detection radius. Okay, so the approach I used was to use density estimates from the aerial survey data to estimate the detectability of harbor porpoise on the passive acoustic sensors. The way that I did this was to take the aerial survey data, use that to construct a density surface. So that's basically a heat map of harbor porpoise density across the study area. And from that heat map, I then extracted the density, the estimated density of harbor porpoise at the location of each passive acoustic instrument. I took those estimates along with the CPOD records, so that's the number of clicks that we heard, and put all of that into a Bayesian model. I used a Bayesian model because you can include data with all of its uncertainty and propagate it all the way through to your final estimate. I also used some values from the literature to parameterize that model. And from that, I estimated detectability. So the visual and passive acoustic data that I used were collected on 3 days in 2013. So these are heat maps of harbor porpoise density on those 3 days. You see that, so the more red means is more harbor porpoise, and you see that they're fairly patchily distributed. Some of that is real because they occur in little clumps, little groups, and some of it is probably observation error. We probably missed some groups. Definitely missed some groups. And then the passive acoustic data from those same 3 days is shown here. So the triangles are, each of the triangles is a passive acoustic sensor, and then the size is the number of clicks that it reported. So that we can then pair these data and plot them against each other. So here I'm showing the density of harbor porpoise on the x-axis that comes from the aerial survey data, that heat map, and then the number of clicks or the porpoise positive seconds on the y-axis. And you see that they increase together, kind of. There's a lot of noise going on in that data. But this is information this does tell us something about the detectability. So we put these data into the Bayesian model along with what's called a prior distribution for the detectability. So this is just showing the whole range of possible values of detectability. I used what's called an uninformative prior. So I said, I don't think any of these values is more likely than any of the other values going into this. With the addition of the data, that paired data set, we then get out of the model what's called a posterior distribution. So this is sort of our answer. And that has a median value shown by this bar and a distribution in the lighter gray. So we did get an estimate of detectability. Breaking that back down into our conceptual framework, so going back to this idea that detectability is actually the product of two things. So we can plot the values of detectability that are consistent with different probabilities of clicking or effective detection radii. And that's what I'm showing here. So this is the effective detection radius in meters, ranging from 0 to 100 meters. And then on the y-axis, the probability of clicking going from 0 to 1. The black line is the median estimate of detectability. So if you took that estimate of detectability and broke it down into its two component parts, that's the median estimate. And then the shaded area are all of the values within the 95% credibility interval for the estimate of detectability. So what this means is that if we had more information, for example, if we knew that we had a small effective detection radius, we could say, oh, that means we have a high probability of clicking. And similarly, if we knew that we had a large effective detection radius, that would correspond to a low probability of clicking. So now that we have an estimate of detectability, we can use this to estimate abundance using passive acoustic data, which I did here. So this is showing 4 months of passive acoustic data, September, October, November, and December. The purple triangles are the passive acoustic estimates of harbor porpoise abundance. And because we flew aerial surveys in October, I'm also showing the visual estimate of abundance from that 1 month. And you'll notice that the error bars around the acoustic estimates are much larger than the error bars around the aerial surveys. That is because the way that I estimated detectability basically took all of the uncertainty in the aerial survey estimates and propagated it through into the acoustic estimates of abundance. So the acoustic abundance estimates have error from both the visual and passive acoustic surveys. And because we only had 3 days of surveys, it's a limited data set. But this is pretty cool because now you can estimate abundance at times when aerial surveys weren't conducted. So you can get a longer time series. And so here I plotted that from September to January daily estimates of abundance based on the passive acoustic data alone. So in this chapter, I think I showed that paired visual and passive acoustic surveys can be used to estimate the detectability of harbor porpoise around a passive acoustic sensor. And based on talking with some colleagues who have tried measuring this experimentally, we're finding that our values are consistent with experimentally measured values. So that's cool. But in order to actually apply this method, right now there's so much uncertainty in the estimate, we really need more paired surveys in order to get a better estimate of detectability in order to use this method. All right, on to chapter 5, which is about designing passive acoustic surveys. So the motivation for this work is that statistical power to detect trends in abundance for marine mammals tends to be pretty poor. So for example, using visual surveys, a 50% decline, so a decrease of half the population, would not be detectable in almost 80% of dolphin and porpoise populations given the current frequency and methods used to monitor those populations. And so passive acoustic monitoring is a promising alternative to that, but we need guidelines to optimize the statistical power to make sure that we can actually get more information out of the passive acoustic surveys than we get out of visual surveys. So the main question for this chapter was, how many sensors do we need and where should we put them in order to be able to detect changes in the number of harbor porpoise? The approach I used was a simulation approach. So I simulated potential passive acoustic monitoring networks and then evaluated how they would perform. So what their statistical power would be under different scenarios of changes in the population. Just to review really quickly what statistical power is. Statistical power is the probability of correctly rejecting the null hypothesis. So in this case, our null hypothesis is that the population is stable, and our alternative hypothesis is that the population is changing. Typically, statistical power increases with sample size, so the more data you have, the higher your statistical power, and also with effect size. So for example, you're more likely to be able to detect a 50% decline in a population than a 10% decline. And conventionally acceptable power, so in the literature, typically acceptable power is above 0.8. So you want to have an 80% probability of correctly rejecting the null hypothesis. The methods for this chapter are going to sound pretty familiar to the previous chapter, except this time on a larger temporal scale. So here I'm showing a heat map of harbor porpoise density estimated using about 100 aerial surveys collected over a 13-year period. So this is just much more data than the last chapter. It's on a decadal scale rather than on a daily scale. And that's why the surface is so much smoother. It's it's much more of a mean estimate. So you see we have this density hotspot again in the northern part of the bay, which is where we deployed our sensors. So again, we had 11 CPODs deployed in the fall of 3 consecutive years. So again, this is a paired data set, just on a much larger time scale. We can use this paired data set to quantify the relationship between the average density and the click rates observed over a whole season. So here, this is harbor porpoise density on the x-axis, and this is the log of porpoise positive seconds on the y-axis. And I used a generalized linear model to describe this relationship. Again, it's much smoother and much cleaner because it's much more of an average. But using this relationship, what that allows us to do is to simulate the collection of passive acoustic data at any location in the study area. So even though we didn't have any acoustic observations at a density of 2, we can use this model to say, oh, we expect the value to be here. So I explored a few different aspects of survey design in this chapter and simulated some different things. So one of the things I played with was placed in the environment. So one way that you might put them in the environment is just randomly. You sprinkle them throughout your study area, and that's shown here. So the little black dots are simulated sensors. Another way you might do it is using what's called a stratified design. So as I mentioned at the beginning of the talk, harbor porpoise are more abundant in shallower water. So you might say, oh, I'm going to put more sensors in the shallow water area than in the deep water area, and that should increase the precision of your estimate of abundance. Another thing that I tried was using what I'm calling a scaled design. So because I have that density surface for the study area, I tried placing the sensors in proportion to the underlying density of harbor porpoise. And you'll notice none of these are a gridded design, and that was just for computational efficiency. But if you were actually going to implement this, you'd probably want to put the sensors in more of a grid. Okay, another thing I simulated were different possible responses to change in the population. So how do the animals respond to a decline in the population? So here's our study area, got a little harbor porpoise all around it. One way they might respond is what I'm calling a uniform decrease. Might just decrease by 50% everywhere throughout their range. So sort of the whole density surface drops down. But another thing they could do is to converge to core habitat. So I'm calling that a range contraction scenario. So in this scenario, as the population declines, they abandon this lesser quality habitat at the edges of their range and sort of contract to the northern part of Monterey Bay. So these are both, I'm showing 50% decreases in the population in both cases. These are just 2 ways that they might respond. Okay, so to go through some of the nitty-gritty of the methods details, as I described, I used a generalized linear model to relate the visual survey estimates of abundance to the passive acoustic data, and that allowed me to simulate the collection of passive acoustic data, given some number of years, number of moorings, percent change in the population, design of the survey, and simulated response of the animals. I used 10 years for all of my simulations, but the combinations of all the possible combinations of these things resulted in 600 scenarios, and I ran each of those 1,000 times each. After each run, I evaluated the simulated data set to see whether a change in the population was detected. And I'm defining that as a p-value less than 0.05 for the year effect in a mixed effects model. And finally, after running all 1,000 runs of each scenario, I then calculated the statistical power as the proportion of those simulations in which a change was detected. And I only considered the p-value and the sign of the covariate for that. So this is a binary, like was a change detected or not. I'm not considering the magnitude of the change. So here are some results from these simulations. So on the x-axis is the simulated percent change in the population, and on the y-axis is the statistical power. The different colored lines are different simulated numbers of sensors, ranging from 10 to 100 sensors. And I'm showing this black line at 0.8 to show you sort of where conventionally acceptable power is. So you see that even with as few as 10 sensors, you'd be able to detect in this simulation, you'd be able to detect a 50% decline over 80% of the time. So you have sufficient power with as few as 10 sensors in that scenario. And these, I should have said, the sensors were placed randomly, and I simulated a uniform decrease in the population in this graph. But if you wanted to detect a smaller change, like a 25% change, you'd need more like 75 sensors to achieve adequate power. So I'm going to focus in on that and talk about sort of that point in the graph a little bit more. So 25% decline with 75 sensors. So now if I fix that, if I say, okay, I'm only looking at the 25% decline and 75 sensors, that allows me to compare the different designs and the different responses. So here, the different possible survey designs are in the rows, and the different possible responses to change are in the columns. And then statistical power will fill in this space. I'm going to show adequate power, so power above 0.8 in green and power below 0.8 just in white. So for the uniform case, so this is when animals decline uniformly everywhere. In our simulations, the design didn't matter. So the power was the same regardless of design, which is not what we expected. We expected that the more complex designs, the stratified and the scaled designs, would have higher power to detect change than the random scenario. So it's probable that there's basically something missing from the simulation of the data. There might be some other source of error that we're not adequately accounting for in generating the simulated data. On the other hand, when I looked at a range contraction scenario, so this is them all contracting to the best habitat, power did vary with the survey design. So while the power was 0.87 in the random case, when range contracted, it was 0.72, so no longer adequate. And only the scaled design achieved sufficient power, although 0.79 is pretty close. So that kind of highlights how if you assume that your population is going to decline uniformly and you calculate statistical power based on that assumption, but the population does something different, that can really mess with your ability to detect trends. So it's important to consider the spatial patterning of the animals and of their response when designing surveys. And just to show the 3 different designs, so this is a 25% decline where the animals contracted to core habitat, varying the number of sensors, and then power on the x-axis. You see that there are differences between the 3 different designs, but what I wanted to highlight is that the number of sensors is really more important, has a bigger impact on increasing power than the design. So in summary, passive acoustic monitoring may have greater power to detect trends in abundance than traditional visual survey methods. So for example, for harbor porpoise, visual power to detect a 50% decline is between 0.14 and 0.33, whereas the acoustic power that I simulated with as few as 10 sensors was above 0.8. And with 75 sensors, you could detect declines of smaller magnitudes, so like a 25% decline. So I think what this chapter showed is just that the assumptions about spatial distribution and response affect our estimates of power to detect trends and are important to consider when designing surveys. So some conclusions on the dissertation as a whole. I think what my dissertation showed was that long-term passive acoustic monitoring for harbor porpoise in California is feasible and that it could provide very valuable data. In chapter 4, we really need more paired survey data to make that method of estimating detectability applicable on a broader scale. Again, I only had 3 days of paired surveys. In chapter 5, I think that statistical power simulation could be refined further. Again, figuring out what's going on with the same power across different designs. And then I'd really like to extend that simulation to other species and regions and do that as part of the postdoc. And finally, I think, this is promising, but we really need a detailed comparison of the costs and benefits of both aerial surveys and passive acoustic surveys. So again, when I said that aerial surveys have low power, that's given the current frequency and extent of those surveys. So we really need to evaluate, okay, which method is going to give us the most bang for our buck in terms of statistical power. And I think that this is going to continue to be a topic of active research, particularly since there have been marine renewable energy sites proposed here in California in the nearshore environment, which is where harbor workers live. Such a nearshore environment, putting a wind farm in the middle could really impact their populations, and I think that's going to drive further methods development. And I think it's really an opportunity to start on the right foot and try to employ passive acoustic monitoring. So with that, I'd like to thank some of the people who have made this possible. My committee members, Jay and Peter, also Karen, Jules, and Bryce. Thank you all so much. I'd like to thank Karen and Jay in particular. I've worked with both of them for about 7 years now, and I've gotten to spend time with both of them at sea, and I just value you both so much as mentors. I've learned so much from you. Jules, thank you for teaching me about acoustics. And Bryce, thank you for teaching me about statistics. Elsa couldn't be here today, but she is my upper campus committee member and just had really insightful questions coming from an ecology background that really pushed me in the right direction. I'm a better scientist because of all of you, so thank you. The Scripps faculty in general are amazing, and I'm so lucky to have been able to take classes like biological oceanography and even things I didn't think I would like physical oceanography. Still not that like, but I feel like it's good for you, yes. Just the range of expertise and the generosity that the faculty here have in terms of sharing their knowledge with students is just really impressive. I felt like I could always ask someone for help, and they were happy to meet with me. The grad office is amazing. Gilbert, Denise, the Maureen's, and Adam, in just advocating for us and supporting us, and I've really valued them. I'm lucky to be part of 2 communities here in San Diego, both the Scripps community and also the Southwest Fisheries Science Center. Thank you so much for keeping that connection strong and for valuing the contributions of grad students to the Southwest Fisheries community. Within the Marine Mammal and Turtle Division, I was sort of part of the California Current Marine Mammal Assessment Program. So I just wanted to thank Elizabeth, Jim, Alex, Mark, Jeff, and Shannon for bringing me into that group. It was really nice, having an office up there and being able to just stop in and ask questions. People who have been working on this data for decades. They're really helpful. Also at Moss Landing, Jim Harvey, the director there, made it possible for me to conduct field work out of Moss Landing. John Douglas taught me everything I know about putting things in the ocean and was just so patient and so kind. And Diana Steller coordinated all of our scientific divers who were just amazing. I was lucky to be funded by a variety of sources in grad school, including the National Science Foundation Graduate Research Fellowship Program and the Interdisciplinary Graduate Education and Research Traineeship. Most of the field work for this dissertation was funded by the California Energy Commission, a grant that I wrote with Karen. And then I was also personally supported by the Sussman Internship, the Tegner Fellowship, and the Achievement Rewards for College Scientists Scholarship. And these things allowed me to travel to conferences and to sort of do side projects that I wouldn't have been able to do otherwise. My favorite thing about Scripps is the grad student community here, and I miss all of you so much. I want to start with this, what we call the Southwest Aggregation of Graduate Students, or SWAG. So these are all students who have advisors both at Scripps and at NOAA Fisheries and have sort of come together to form a lab and support each other. They're all just such amazing mentors and people. The Franks Lab, thanks guys. Yeah, so I became Peter Franks' student in my second year, and he just welcomed me right into the lab. Thanks especially to Jess and Bryce for all of your help. And the 2012 BO cohort. I came in a cohort of 5 women who are all amazing, strong women. We all do very different things, but I think we've all pushed each other to be better scientists. And the 2012 cohort in general. My friends. who are willing to dress up as a flock of flamingos. These are hand-sewn flamingo legs. So Sean, Jess and Max, Julian, Peter, Nick and Caitlin, Jess and Spencer and Jenny and Josh and Mads and Brian and Sarah and Amy. Thank you all for all of the game nights and the family dinners, the Friendsgivings and the Easter egg hunts. Grad school would not have been the same without you. There are a couple of women who have played a really important role in my life, including Amy Van Sice, who couldn't be here today because she's on a cruise. Amy and I kind of went through grad school together. We traveled a lot together at conferences, and just it felt like we were a team. And I was just so grateful to have her by my side throughout this process. And my roommate Liz, Liz and I met in 2008, where we sang karaoke in the Florida Keys. And I moved in with her earlier this year, and I couldn't have asked for a better place to live while finishing my PhD. She's just such a warm and generous person. And Sarah Lurch. I've had so many good adventures with Sarah, so much fun, and I can always count on her to be a voice of reason, a voice of support. I value you so much. And actually, we went on a kayaking trip last month, so I'm a month before defending, and we went up to Alaska for a week kayaking. Thank you, Sarah. I'd like to thank my family, so many of whom are here today. Thank you guys so much for coming and for supporting me. My parents, Galen and Eric, my brothers, Mars, Austin, and Oslo, my grandmother, Bernadette, and my aunt, Aileen, and my grandparents, Jake and Penny. Thank you all for recognizing that this is an important moment in my life and being here today. I'd like to thank my cat Fern and my dog Juniper. Juniper will be at the party later if you need some fluffy cuddles. And also Harbor Purposes everywhere. They give me such joy every time I see them. So with that, thank you all.
Fun. All right, questions? Yeah, Lisa.
Very nice job, Eiren.
Thank you.
I want to go back to your detectability chapter and ask you a question about that value, that metric that you calculated. Although you said that you would like to see more paired surveys, I think it's a potentially powerful metric because of the potential for transferability, and I'd like to hear what you think about that possibility for transferability in two ways. First, you showed a nice map of the global distribution of harbor porpoise. So what are your thoughts about using that detectability parameter for harbor porpoises outside the Monterey Bay area?
Okay.
And then second, what are your thoughts about using that detectability metric for the other species that make echolocation clicks that are almost close to identical to harbor porpoises? You showed Kogia and Cephalorhynchus, I believe.
Okay, so first question, applicability outside of Monterey Bay. I think you could do that. I think it would not be ideal, I guess. I think you can do whatever you want, right? But especially thinking about different oceanographic limitations. So for example, in the Baltic Sea, when people did playback experiments there, where they played an artificial sound source back to the sensors to look at how far away from the sensors it was detected. And they actually ended up fitting different detection functions for, I think, each month and for day versus night, because of the stratification there really affected the propagation of sound. So I would say I'd be comfortable using this for California and maybe up into Alaska, but then you have to start thinking about, are those really the same animals? You know, they tend to get bigger the further north you go. So I would not recommend it. And I'm not necessarily recommending that we do more paired surveys. I just wanted to say, this is a way you could do it. You could measure it directly using playback experiments or similar, or you could do it using paired surveys, but you'd need a lot of paired surveys to get a good estimate. Your question about applying to other species, I think behavior is, we just don't know enough about behavior, especially Kogia, correct me if I'm wrong, tend to click more like sperm whales. So as opposed to harbor porpoise that are sort of echolocating continuously and sweeping their beam, my understanding of Kogia is that they're more in very regular series. And I don't know if they have the same beam pattern. I don't know anything about Cephalorhynchus. Dahl's porpoise are very similar. I think they have similar behavior, so possibly. But I think they're louder. So that's also something. Yes.
When you paired your CPOD data with your visual surveys, you interpolated linearly to create that heat map?
So in one chapter, I used a technique called objective mapping. And in the other chapter, I used spline on latitude and longitude, basically.
So do you think your results would change if rather than interpolating your data to a grid, you fit your data with distance from the CPOD, assuming that the CPOD picks up on all of the animals that are within the radius rather than having a fixed value?
So you would bin the data around the CPOD and then say, count up the total number of animals that were in that. Oh, I see what you're saying, because I'm using just a single point. I think in some of the cases where in the chapter where I used objective mapping and the heat map was pretty patchy, I think, yeah, it could make a difference there because the density changes so rapidly. In the other chapter, that surface was so smooth, I don't think it would make a difference.
In the power analysis stuff, does the density, the uncertainty on the density estimates factor into that? And if so, had greater confidence intervals on the densities, would that be one of the sensors you need to detect the decline?
If your aerial survey density estimates were more,
Not like your density estimates. So you have in your power analysis, you say I need 50 sensors to detect the decline. Does the uncertainty on the time series they've had with the density, the acoustic density, the uncertainty factored into that power analysis as well?
oh, it does in the sense that, so I didn't use any of the results from chapter 4 in chapter 5. They're pretty separate. But I did include sources of uncertainty like that in the simulation framework. So I didn't use, for example, fixed values of density. I said I pulled densities with some mean and some error around that, and the same with the clip rates. So I showed that generalized linear model that described the relationship. So if I wanted to simulate a sensor at a density of 2, I would say, okay, the median value is here, and then there's this uncertainty, so pull a value from that distribution.
The confidence doesn't.
Yeah.
And kind of confidence.
I think that what she didn't catch was that you didn't use that correction factor for going from aerial to acoustic in your simulation of detecting change, because in detecting change, you're just interested in relative abundance, right?
Yes. We should talk about that.
And
I have a question. Hi, you did, I think it was chapter 4, you showed your density estimations over a series of months. And it looks like there was some sort of pattern in there. Do you know what's going on there?
Are you thinking lunar?
Well,
Let me guess.
I am thinking lunar, but I didn't need to look at it fast enough to see exactly where they fell. Did you?
I haven't looked at that, and I think again, you know, the confidence intervals are so large. I don't know if that's real or not. But that's something to, look into. And Wayne had a question, sorry.
You know, I was gonna ask the same question.
Oh.
It didn't look random, and I was wondering whether it could have been
Yeah.
here, or it could be that the animals are just, I was curious
Yeah.
to get a wild guess.
Yeah, the other thing I didn't really talk about is that in a lot of this, I assumed what's called a closed population. So I assumed animals aren't coming in or out. So that could also be, so they could be following a ball of fish, or oceanographic conditions could be changing the detectability.
I'll add, having been in the plane for most of those additional 26,000 kilometers, we kind of view that in this region, porpoises, I call it the blob, that they do, that since the study was really limited to North Monterey Bay, sometimes they just go south, they go out past Santa Cruz, and you usually find a ridiculous patch of really crazy porpoises like her heat map showed, and most of the time it's in the water, sometimes they go out. And I think it would be interesting to look at other oceanographic. I suspect, if anything, it has to do with upward relaxation. Meetings rather than understand. That's just what we've talked about a little bit. Don't think.
So I'm thinking about them from the point of view of the fish. And I'm wondering, you analyze your statistics on a more point-by-point basis, looking at, say, fractional occupancy of time, but I'm wondering if there are correlations in the second-order statistics that would indicate, say, increased prey densities where they were actually foraging.
Right, and I think that's what Anna's question is getting at, because some of her work has shown, correct me if I'm wrong, changes in foraging with lunar cycles.
Yeah.
Okay, sorry.
To that.
Okay.
But is that a useful?
I think it just makes it messier, right? You know, then it's like, well, if detectability is changing with the moon.
The moon's pretty predictable.
It's true. If you could get get enough measurements to make lunar specific estimates of detectability.
No, but I'm wondering about the temporal variability.
Yes, I think that's something that we just don't know very much about.
And they're solitary pretty much, right? But they don't chirp or whatever, but are they also communicating with each other?
There is like one paper that suggests that they communicate with each other using their echolocation clips. I don't know, Jake, if you have opinions on that, but there's limited evidence that they communicate with each other.
So do you think they're more successful hunters because they're solitary? Because if you imagine a group of animals that were foraging and they were actually queuing off each other's success, and prey densities were patchy, maybe, you know where I'm going.
Yes.
So I'm sort of asking you to frame this in terms of the ecology of their prey.
Okay, so I think that I don't want to anthropomorphize, but they've sort of made an
Do it all the
evolution.
time.
They've made like an evolutionary decision, sort of, that getting eaten by killer whales is such a risk. Like, it's not worth it to be social. It's not worth it to cooperate or to hunt together. They've sort of taken this track of like, all right, we'll just be as quiet as possible, make just the amount of noise that we need to make, and to avoid getting eaten. So I think
I often tell students that humans were not usually worried about being predated on our way to the supermarket.
not usually. Oh, hi, Mom.
Don't make her cry.
You have a what question? Methods question? Okay.
So when I see all the work that it takes to go out and deploy your monitors, and I imagine there's a lot of other scientists out there putting monitors, is there any cooperation where you put in that, stack them up and like listen for whales and dolphins and put all the instruments into one?
That is something we need more of for sure. That's definitely a problem. Even within institutions, we tend to do our projects separately, not collaborate enough. But yes, that would work.
Somebody might be doing it.
Oh,
helped me answer.
Jules?
No, John.
What are you putting on your harps?
What was the question?
Oh, the question was just about putting multiple types of sensors on single mooring.
We did a experiment
That's
where
true, we did.
We had C-pods and harps together. You didn't present it, but
That's
did you work
true.
on that data?
I did 3 years ago.
We're not
Yeah,
gonna
you're right. So John and I did put a C-pod on a harp to basically, so one of the chapters I didn't talk about was comparing a C-pod to a sound trap and looking at how many clicks are recorded on each and what the frequencies are. We also collected data with a C-pod and a harp with the same idea, but I ended up using the C-pod sound trap data because the sensors were closer together. So I thought that was a better comparison for what I was looking at. So, yeah.
Questions from the peanut gallery? All right, well, thank you again, Eric. Great job.
Remind you that there's going to be a party over at Surfside in a few minutes after we've had a chance to grill her for a while. There's also a party at my house tonight, so anybody who would like to come to that, please contact me. I'll be at the after party, and I can get the address and stuff like that. But 6:30, my house tonight.
Great. See you soon. Those are good questions.
Yeah.
So people understood it.
Yeah. Yeah.
Mom asked the question. Awesome.
Yeah.
Her to become the assistant.